We start chapter 16 today. In 16, we're getting down to the molecular basis of inheritance, down to the DNA, basically, and the RNA. Okay. Remember, Watson and Crick are the ones that get credit for the shape of DNA, deoxyribose nucleic acid, even though we talked about Rosalind Franklin. We'll talk about her too. Without her, these two guys wouldn't have gotten anything done. But you know how that goes. We already talked about that. But anyway, they came up with the shape of DNA. Okay. And once they knew the shape of DNA, they could start doing other things. The thing to remember about DNA is the blueprint to build you. And every time a cell reproduces, it has to make that same DNA so that the two new cells have the same DNA as the one cell that it came from. Okay, that's hereditary. And again, Mendel did all that without knowing anything about DNA. He knew that there was factors, and now we know their genes and all that stuff. Okay, so the DNA is what is responsible for the development of you. Okay, all the biochemical processes, all the reactions that occur, metabolism, catabolism, anabolism, we talked about all those. That's all from enzymes and proteins. That's DNA. Your anatomical, in fact, you got 2 arms and 2 legs and ears and a brain, and how your kidneys work and all that stuff. It's all in your DNA. And they say to some extent your behavioral traits, that's an argument. They say, are you who you are because of your genetics, or are you who you are because of your environment? That's a debate, and I don't think it'll ever get settled. Okay, so there's what DNA looks like at the microscopic level. You've got your sugar phosphates, and then these are your nitrogen bases, your A's, T's, C's, and G's. Okay, so we know the DNA is copied they call it DNA replication. It's when the cells are going to divide, or if you need some material, like you need to repair some cells, okay? It'll go in there and open up, the DNA will open up, and it'll make a copy of whatever gene you need, and then it'll close back up again. Now you got your copy, and you can make your protein. That's usually done by that messenger RNA if you need to make copies to make genes or proteins. But for replication, it's just making exact copies of the DNA so the 2 new cells get the exact same material. Okay, so we talked about Thomas Morgan when he talked about the fruit flies, and he's the one that showed that genes are located on chromosomes. Okay, he also figured out chromosomes are made out of 2 things. They're not just DNA, they're also a protein called chromatid. So when you look at a normal cell, the DNA is really skinny and stretched out. You cannot see it with a microscope. But when the cell gets ready to divide, what happens is these chromatid proteins make the DNA, instead of being really long, it coils down into really small around these little proteins. And that's why you can actually see it with a regular microscope. The little X's, the little chromosomes, those are chromatid and DNA. Okay. He figured out, and again, Morgan and Mendel, both of them figured out that Mendel didn't know about DNA, Morgan did. Okay. But they figured out this is the basis of hereditary. There's something there's some molecule that's in charge of all that. And they've learned a lot about DNA by studying bacteria and the viruses that infect them. Okay. They've learned that, how the mechanisms work and so on. Okay. So a guy by the name of Frederick Griffin, you can see 1928, it was 30 years till they discovered the actual shape of DNA. But he knew that there was this molecule, and he started doing research with it. And what he found was he would take bacteria. One was caused a disease, pathogenic, and one was harmless. Okay. And he would kill one of the pathogenic one. He would, heat it. That's all you have to do to kill a bacteria is heat it. And he found weird things when he put this one and this one together, it became pathogenic again. And so he's like, there's got to be something that's inherited to turn it back on again. Okay. So what he did first is he gave him, to prove his point, he gave a mouse, you know what? I'm going to show you this instead. He calls it transformation, and it's something that happens with the DNA, but this is a better, I could babble on, but this will explain it better. Okay, so he took some sort of bacteria that caused a disease, okay? So we'll say the S cells. All right, he gave it to a mouse, the mouse died. So he knew it was pathogenic, it gave it some disease, and the mouse couldn't survive. Then he found a bacteria that was similar. See, this one has like a coating on it. This one does not. Okay, so this was the non-pathogenic one. Gave it to the mouse, nothing happened, all is well. Then what he did was he took this and he heated it. And basically he probably destroyed this coating. Okay, gave it to the mouse was good. So now he knows that this mouse, this whatever this diseases is he killed it. Okay, the mouse is good. But for some reason, when he put the two of them together, the heat killed S cells, and the normal ones, the ones that don't cause anything, all of a sudden, he gave it to mouse and the mouse died. So what happened? The harmless somehow activated this. So we knew there had to be something that was, when it divided, was carried on. And you can't see down here, but it says the living S cells again, they look like this. Okay, so this was Griffith's experiment, and he found that there was some sort of factor that could reactivate stuff after they'd been destroyed. Okay, there's other people, Avery, McCartney, McLean, they did some transforming DNA. Okay, at this time, this is 1944, so you're still 10 years away from the structure of DNA. A lot of people still weren't real, like, really, okay, maybe, I don't know. Okay, is there really this mirror, this big molecule that does all this? So it was still skeptical. So they had to do some more evidence with bacteria and with viruses to prove their point. Okay, they did a lot of studies with viruses to infect bacteria. See, a virus in itself isn't alive. It's just a little piece of, they say DNA, but I think it's more RNA. Okay, it's a little piece, and for it to work, it has to get inside a bacteria, and then it's alive. Okay, so when a virus, they're called bacteriophages or a phage, and what it is it's a piece of RNA that needs to get inside of a bacteria in order to work, and they use it a lot in molecular genetics research. They do a lot with these because they're easy to use. They reproduce quickly, and when it's done, it's easy to destroy them. Okay, but a virus is DNA or sometimes RNA, and it's enclosed by a little protective coating. It's just some protein, kind of like you saw on the slides with the mice. There's like a protein coating around them, and it protects them, keeps them together. Okay, so what happens is the phage head, if you want to call it here, this is the virus, and you have your little DNA or RNA, whichever one it has a little tail. It comes in contact with a bacteria and it attaches to it, and then this part, the head, is allowed to travel into the bacteria. Once it gets in the bacteria, then it takes over the bacteria. Okay, now it has a vessel to keep it alive, and now it starts taking over and doing whatever it wants, basically. Okay, two other people, Hershey and Chase, they showed that DNA is genetic material of this phage. They called it the T2. Okay, and they designed an experiment showing that one of the two components, either the DNA or the protein, enters the E. coli. Okay, so they had to figure out which one is it. Is it the protein that's the hereditary stuff, or is it the DNA? At that time, they didn't know. Okay, so it gets into an E. coli. E. coli is a really common bacteria. Okay, you've probably heard of E. coli poisoning. We have E. coli in our intestines. There's a lot of stuff, but again, it's a common bacteria. They work with it a lot. So they concluded, they injected DNA of the phage, and they wanted to figure out, is it the DNA or is it the protein that causes, this taking over of the cell? Which one is the hereditary? So they labeled, okay? They labeled the DNA and they labeled the protein. And they mixed them all up. This is our centrifuge. This is our mixing, good old blender. Mixed them together. They centrifuged them, and they ended up with a pellet. Okay, this is the radioactive stuff is here, and then the pellet. Okay, they found the phage protein was in the liquid. Okay, oh, sorry, we can't see it so much. When they did the radioactive material, the DNA made it radioactive. In other words, they put like a tag on it so it fluoresced. Did the same thing, mixed it up, centrifuged it, and they got their pellet. They found out the pellet, which is the bacteria, had the DNA. So here, the pellet, which was a bacteria, did not have the protein in it. Down here, it did. So they knew that because the DNA was radioactive, it's the one that got inside the bacteria and changed it. So this is when they figured out the DNA was the hereditary stuff and not the protein coding. Because at the time, they didn't know which one it was. So this was the experiment that proved that DNA was the one that had the hereditary information. Okay? So now they know that DNA is the genetic material. Okay? They know a few things about DNA. They know it's made out of nucleotides, sugar, phosphate, and a nitrogen base. Okay, and they know it's a polymer. In other words, there's lots and lots and lots and lots of them. Okay, so Chargaff, he's another guy. He started looking at the composition of the nitrogen bases. Okay, so he'd take DNA and he'd figure out the percent of adenine, the percent of thymine, the percent of cytosine, and the percent of guanine. Okay, the evidence of the diversity, made it more credible because they had this genetic material now. It was there was more and more experiments doing all the time. So now it was like, okay, we got it. DNA is the genetic material. We know that. Okay, and what he found when he did his experiment is the amount of adenine and thymine percent-wise was always the same. Cytosine and guanine were always the same. So now they know when you look at a DNA molecule, the two nitrogen bases that go together, A and T, C and G. Okay, so if you look at a DNA, you have a phosphate group, a PO4 group, and you have a sugar. The sugar is deoxyribose. And these are the sides. Okay, and then the bases. And here you have your bases, and you have your two nitrogen bases that are double strands, and then you have single strands. Well, a single and a double go together, and a single and a double go together. And they always go in the same order. That's why the code for the protein is always the same, because if thymine sometimes goes with cytosine or sometimes goes with guanine, then it's going to change the code. So A and T always go together, C and G always go together. Okay? And Chargaff is the one that figured that out. Okay? He figured out the base composition of the DNA varies between species. Okay? But when he did his numbers, the A and the T bases were always equal, and the C and the G. So if A was 20%, T was 20%. And that would be 30% for these two guys each. And that would give you 100% of your bases. So he did lots of numbers, and he always found that the A and T numbers were the same, and the G and C. And it equaled 100, but these two equal each other, these two. That's why they know they pair together. Okay? And they didn't really understand. I mean, they knew it was there, but they didn't really understand the process until they discovered the shape, the double helix. Okay? So now we know the DNA, they finally accepted, yep, DNA is the genetic material. This is the stuff that causes traits to be inherited. And we know that we get half of our traits from mom, half from dad, all that stuff. But they really wanted to know the whole story. Maurice Wilkins, and then here's poor Rosalind Franklin. They were the ones that you were using X-ray crystallography they were photographing DNA. All right. Franklin was the one that produced the picture of DNA using that technique. She's the one, but again, remember in her time of day, women scientists are like, yeah, whatever. So her boss stole the picture and gave it to Watson and Crick. Okay, there she is. And this is what she found. Using X-ray diffraction, she was the first one to photograph DNA. Then she did the math to figure it out. Watson and Crick, they took the image, and I guess they were more free thinkers, and they came up with the idea of the shape of DNA. So did Franklin, but she did it mathematically. They did it through, I guess, hypothesis and stuff like that. So by looking at that X-ray image, they were able to deduce the width of the helix and the spacing and so on. And they're the ones that figured out that the nitrogen bases were on the inside. Again, so did Franklin, but she was a female. She was a little slower. And it wasn't really fair that they stole her photograph. Okay. But the photograph is the one that really set up the double helix, showing him there was two strands. So it's like a spiral staircase. Okay. So again, here's DNA. The sides are sugar and phosphates. It's always the sugar that the base is attached to, and it's always ATCG. Okay, so that's the DNA. This is the structure that we know DNA as of now. Okay, and that's how they always they bond that way. I'm looking at time here. Okay, so Watson and Crick build models of their double helix to conform to this X-ray diffraction and use a little bit of chemistry. They concluded that the two outer sides were the sugar phosphate, and the nitrogen bases were basically the steps. Okay, so they get credit for it. I think in 1962 is when they won the Nobel Prize. Franklin doesn't even get any credit because by that year she passed away and didn't get a chance to fight. Okay, so they built their model backbones. Their subunits run in opposite directions. You'll see that the 3 to 5 to 5 to 3. Okay, and they're the ones that get the credit. Okay, and that's where we're going to quit for right now.